This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 203, recorded on October 12th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Today, we are in Alexandria, Virginia. We're back in Alexandria. You may remember we were here some time ago. And yes, we are at Ed Niles' house. But Ed Niles is not with us today. With me today from North Central Florida, Rich Condit.
Hi, Vincent. How are you?
Across the room from me.
Yeah, here we are, face to face again.
Ed Niles' living room.
Yep.
After study section, just like last time, right?
Exactly.
And we are here because we have a guest today who we're going to speak with.
Wait a minute.
What's that?
The weather.
Before the guest, even.
Yeah.
Okay, weather in Alexandria.
Beautiful day, sunny blue skies.
It is.
Fall's just starting, 65 degrees Fahrenheit, whatever that is in Celsius, I don't know.
I don't have my phone with me,
Beautiful
so I can't give you the Celsius. Fairly nice day here. I have no idea what it's like in New York. It's probably similar. Okay, our special guest today is from the National Institutes of Health, NIAID, National Institute of Allergy and Infectious Diseases, the virology branch in the Division of Microbiology and Infectious Diseases. He is a program officer for alpha herpes viruses and pox viruses. It's Mark Challberg.
Hi, Vince. Good to be here.
Welcome to TWiV, Mark. We see you all the time, Rich and I, at study sections, and now we get to talk to you. That's great. Thanks for doing this with us.
No, my pleasure.
So we just finished, Rich and I just finished study section, and we thought that it would be a nice thing to chat with you about your whole life. Starting from the beginning.
By the way, study section, I don't know if we've actually really talked about this in detail, but for the listeners who don't deal with this every day, what that is it's a panel of scientists who do peer review on grants. So when a scientist submits a grant to NIH asking for money, that goes through a bunch of administrative stuff that we'll probably hear about a little bit today, because Mark is part of that process, and winds up, along with a bunch of other grants, in a peer review process that involves getting together a group of 20 or so scientists to evaluate the scientific merit of these grants and then pass them on with those rankings to the NIH administration for determination of whether or not they're going to get any money. So that's what study section is.
So I wanted to start from the beginning with you, Mark. Let's just start where are you from? You have a Southern accent, right?
I don't think so. I hope not.
It's not Southern? What is it? Is it Brooklyn?
Well, I actually grew up overseas. My parents were missionaries. Grew up in South Korea.
Okay.
And I left Korea when I went to college, and I went to college in Maine.
Wow.
So it's hard to see a Southern accent coming.
What do you think? You don't have any sense of Southern accent in this?
Well, he talks a little funny.
Talks a little funny.
But,
okay.
Must be all the time I've spent in Maryland.
Maybe. That may be
Maryland.
Maryland.
Could be a Maryland accent. So which college in Maine did you go to?
Bowdoin College.
Bowdoin, okay. And
so
then,
you lived in Korea till you were how old?
18.
Wow.
Do you speak Korean? Or did you?
I did, a little bit, yeah.
Huh.
Although I went to an American high school there, so.
Yeah. So in Bowdoin, what did you major in?
Chemistry.
Okay. And when you graduated, were you ready to go on for a PhD?
Yes. And so then I went to Johns Hopkins Medical School, where I got my PhD in the Department of, it was actually, in the Department of Physiological Chemistry at the time.
This is roughly what decade?
Early 70s.
Okay.
And that was with Tom Kelly, right?
No, that was, got my
Paul
PhD with
England.
Paul England.
Of course. Okay.
And then I stayed on at Hopkins to do a postdoc with Tom Kelly.
Okay.
So, and for your PhD, what did you do?
I worked on vaccineovirus.
Wow.
Vaccineovirus DNA polymerase.
That's why you guys are friends, right?
I didn't know. I didn't know Mark then.
No.
I didn't meet Mark until way into the herpes era. I don't think, which we'll get to. But I must say that my very first PhD student worked on a temperature-sensitive mutant that we isolated in the virus DNA polymerase. And to prove that that was a temperature-sensitive mutant in the polymerase, we went so far as to partially purify the enzyme and show that it was thermolabile in vitro. And what that really involved, and this scared me to death, right? Because, I was not a biochemist. But what we did was made just small scale lysates from infected dishes, made lysates and ran them over a DEAE column and eluted stepwise with different salt concentrations using Mark's protocol as our guide, because what he showed is that the vaccinia polymerase eluted at a different salt concentration than the host polymerase, and there's a very clear two peaks. And in uninfected cells, there's only one peak. And in infected cells, there's two peaks. And we took the second peak, showed that that was thermolabile. So thank you, Mark. It was terrific.
You're quite welcome.
It worked like a charm.
So you had purified the vaccinia polymerase by this column that that Rich is talking about.
And a few others. Right.
So then I have a paper here that Rich put up from 1979, Challberg and England, Purification and Properties of the Deoxyribonucleic Acid Polymerase Induced by Vaccinia Virus. So
that would be the one.
Part of it, right? And this polymerase was a complex, I guess, or was it a single polypeptide?
It was a single polypeptide.
Coming right off the column, one polypeptide, right?
Well, after several columns,
yeah.
one polypeptide.
And that single polypeptide has activity by itself?
It has DNA polymerase activity by itself. It's not competent to replicate vaccinia DNA because the way it actually comes is as a complex with two other proteins. But we didn't know that at the time, and we purified it using an assay that didn't depend on these two other subunits, and so we got a single polypeptide. So
at this time, this is 1979. Was this the first viral DNA polymerase purified?
It might have been.
I'll bet it is.
First animal virus DNA polymerase. Certainly phage polymerases had been purified.
Right.
That makes sense. Right. Okay, that makes sense.
So you went into this was a PhD program, and England said you should purify this enzyme, and you did?
Yeah,
that's how it works?
What I did. That pretty much sums it up.
Nice.
Did you infect like HeLas and Spinner or something like that?
Spinner, yeah.
Okay. So a lot of stuff, right?
Yeah.
Fairly large volume.
I think the standard prep involved about 30 liters of HeLa cells.
Okay.
You say in this abstract that it co-purifies with an exonuclease.
Right.
Is that part of the...
That's an activity that's intrinsic to that polypeptide, and it's typical of many DNA polymerases. So the exonuclease acts as an editing function to get rid of misincorporated nucleotides. So
if you took this polypeptide and you added... Well, how did you assay polymerase activity? What was your assay?
Using what was called at the time activated DNA, which was just calf thymus DNA that was incubated with a little bit of magnesium for a while so it could get all nicked up and gaps and so forth. And then the polymerase, sort of the minimum activity of a DNA polymerase is to be able to add a few nucleotides to expose 3' terminal.
Okay. Calf thymus DNA, wow.
So the activated DNA basically is in effect, a template with a primer on it.
Right.
Yeah.
Right.
And that's what the polymerase works on.
And you put a radioactive triphosphate in, and you measured incorporation, right?
Right.
And used a scintillation counter, probably, right? To measure.
Scintillation counter, absolutely.
So you remember this work still?
Oh yeah,
Yeah, it comes right back. That's good.
absolutely.
Oh, that's cool. So then, after you finish, do you want to do anything more before we move on to?
I would just
POSTA?
make one other comment about this polymerase, since
Yeah.
we've talked a little bit about it, and that is, I mean, Mark didn't know this at the time, but it looks like this polymerase is the enzyme that does recombination in vaccinia as well.
Mm-hmm.
So it catalyzes strand exchange, which I think is a little unusual for polymerases. So I think it's weird and interesting in that regard.
And this is the DNA polymerase of vaccinia virus. There's not another one. It's not a family of different polymerases. This is it,
This
right?
is it. And of course, unlike many DNA viruses, vaccinia does all of it, what it does in the cytoplasm of the host cell, so there aren't any other DNA polymerases around there.
That's right. That's right. It makes its own factory in the host cell, right? So you finished this, and you've already told us you stayed at Hopkins to do a postdoc,
Right.
with Tom
With
Kelly.
Tom Kelly, yeah.
And why did you stay there and not go somewhere else?
Well, this is sort of a long story. I actually had arranged to go somewhere else,
Okay.
but my wife was finishing up some postdoctoral work at Hopkins as well, and so I decided after I got my PhD to stick around for a year and just wait for her to finish up. We were both supposed to go out to Stanford at the same time,
Okay.
and so I started working on this project with Tom, and things just worked out so wonderfully well. I just couldn't leave it, so I stayed.
That's good.
Yeah.
Can't beat that. So you went to Tom Kelly. You said, "Could I work for you?"
Yep.
And he knew you had experience with DNA polymerases, and
Right.
that was his interest at the time, right?
Right. Right.
Except he was working on a different virus.
He was working on adenovirus rather than vaccinia
Right.
virus, and he said, "Yeah, I want to get into the biochemistry of adenovirus replication, and you obviously have some experience with DNA polymerases, so yeah, why don't we do something?" And
So at that point, would you consider yourself a biochemist or a virologist?
certainly a biochemist.
Yeah, sounds
Yeah.
like it.
Yeah, I was just very gradually reeled into the world of virology.
So what did Kelly tell you to do?
He said, "Well, what we need to do is get an in vitro system that faithfully recapitulates DNA replication in a cell-free system." And he said, "Nobody's been able to do that, but you got a year. Doesn't matter if it works or doesn't work. Give it a whirl."
And how did he choose adenovirus? Because as it turns out, adenovirus is a pretty weird system to
It
do
was,
that in.
but Tom was working on adenovirus. So.
Okay.
Why is it weird?
Because of the terminal protein priming mechanism.
Okay.
Plus, as I recall, and we can get into this if you want, as I recall, it took a while to figure out that adenovirus actually encoded its own DNA polymerase. I recall that being kind of a mystery.
Right, although that was actually known.
That was known?
That was
Okay.
known, yeah.
So he said, develop an in vitro system. So was the polymerase purified by that point?
No, the polymerase wasn't purified.
Was the terminal protein priming thing known?
No, although it was suspected. And
I remember that starts out with Tony Robinson, working on CeLO virus, figured out that there was a terminal protein, and then Rikosh and Robinson and Willie Russell
right.
took that over and really refined that. But I guess you had some work on that down the road as well.
Well, that's right. So I guess, what I did was to say, as you say, just about that time, it was becoming obvious that the ends of adenovirus DNA, the 5' end, was covalently connected to a protein. And so that's how it comes out of the virion. If you purify adenovirus DNA without proteases, there is a protein attached to each 5' end. So I said, well, that DNA with the protein covalently bound has to be the template for DNA replication in vivo. So we should start with that in vitro. So I just made extracts of adenovirus infected cells and purified the protein, as I say, in a way that did not involve the use of proteases to preserve the integrity of the terminal protein, and use that as a template for in vitro DNA replication. And the first experiment I did worked. It was amazing.
But did you purify the polymerase, or was this an extract?
It was just extract.
Extracts of infected cells.
Infected cells, right.
So you're adding purified adeno DNA. So, and then you got replication of that?
Right.
So how could you distinguish it from endogenous DNA in the extract?
Well,
Maybe you couldn't, right?
well, you could, because you could run it on a sucrose gradient.
Okay.
So adenovirus has a discrete length, and so that was one way.
Okay. So
And also there were several controls built into this, so it only worked with infected cell extracts, and it also only worked if you kept the integrity of the terminal protein on the template.
Okay. And you did that experiment. You cut it off, and you showed
Right.
it wouldn't replicate any longer.
Right.
And so here we have a 1979 paper, Challberg and Kelly, adenovirus DNA replication in vitro. And you did that in that first year?
I did that in about the first week.
Wow, that's great. In the last sentences, the system provides an assay that should be useful for the purification and subsequent characterization of viral and cellular proteins involved in DNA replication. So is that what you did next?
Yes.
So you stayed in Tom's lab?
I stayed in Tom's lab for the next 3 years.
3 years, okay. And did you purify some proteins that were involved?
Purified some proteins. I actually spent more time on using the in vitro system to demonstrate the mechanism of initiation, the protein priming mechanism.
Okay.
So how does that work? Let's lock this down.
So DNA polymerases are known to only add nucleotides to 3' ends that, hybridize to a template. So unlike RNA polymerases, DNA polymerases in general cannot start with a 5' triphosphate and add the next nucleotide to that. Something has to be there already. And so it was always an issue in the case of linear DNA, double-stranded DNA viruses, how DNA replication could initiate at the ends of a molecule. How is that first nucleotide? So the way that adenovirus solved it was to use the hydroxyl group of a serine residue in a protein as a stand-in for the 3' hydroxy terminus of a DNA strand. So the first nucleotide that adenovirus DNA polymerase polymerizes to a protein, or it covalently adds it to a protein. And then DNA replication starts from there.
So the polymerase actually adds the
Yes.
nucleotide to the protein.
Right.
This is called terminal protein, right?
Right.
And that acts in effect as a primer
Right.
for the rest of the replication. So
Right.
you worked on aspects of that then.
Right.
So this was 1979. I guess by then the sequence of the genome was known?
A little after that. I think it came out in the 80s.
And then you could see the coding region for your protein that you'd been studying. Now, adeno is different from POX in that all the DNA replication takes place in the nucleus of the cell, right?
Right. And one of the issues that came up, so it turned out that we did an experiment to determine the size of the protein that is used as the primer, for DNA replication. The idea was to prove that it was the terminal protein that it is attached to virion DNA. Well, did the experiment and it turned out to be a different size. So that was a little bit puzzling and a little bit disconcerting. And so I spent maybe a year trying to sort that out. And what it was the terminal protein. It's the primary form of the terminal protein was quite a bit bigger than the terminal protein on virion, and it just, the way it worked is that protein, the larger version of the protein, was processed down to the smaller version of the protein during the process of encapsulation. So,
Yeah, has that
proteolytic processing during encapsulation.
Is this protein priming of DNA replication? Is that unique to adenovirus, or is that shown up anywhere else?
Well, it's I don't think there's any other animal viruses that do that. There is a bacteriophage that do it exactly the same way.
Okay.
Is it V29, I think? 529?
No. Is it?
No, it doesn't matter.
Yeah.
There is a phage.
Yeah.
Of course,
Cells don't do this, right?
No.
of course, polio replicates with a primer, but it's an RNA virus, right?
Right.
There's a little VPG.
Right,
Well, the
of course.
picornas have VPG on their RNA, and that's a primer for replication.
Right.
In fact, I remember years ago when people worked on the polio problem, they were always comparing the adeno priming situation because it was the only other, besides the phage, it was the only other situation.
By the way, if people go back to Virology 101,
Right.
TWiV 96 is on DNA replication, and TWiV 106 is on DNA replication.
That's right. We did 2 of them.
And we did 2 of them, and we talk about this issue with not being able to replicate linear molecules.
Yeah.
And we talk about the adenovirus DNA replication. So if you're interested in seeing all the drawings that come with it, those 2 TWiVs have slides with them and everything else.
So the reason I mentioned the nucleus, so adenovirus makes its own DNA polymerase, but it probably uses other proteins for replication in the nucleus, right?
I don't actually think it uses any
No.
host proteins. Adenovirus also encodes a single-stranded binding protein.
Okay.
And it is an unusual mechanism for DNA replication, right?
Right.
It's not your standard replication for it because it's this strand displacement
Right.
thing, right?
Just goes from one end to the other, displaces the non-template strand.
So you don't have, there's no lagging strand synthesis.
Right. Exactly.
So it doesn't require all of the junk that might ordinarily be required to do that.
So you displace a strand, you make a new duplex, and then the displaced strand will eventually get replicated as well. I
Right.
think,
It's used as a
Yeah, template.
template. Right.
So there's a double-stranded template, and then there's single-stranded templates. They both work with the polymerase.
Well, actually, it's a little more complicated than that because adenovirus has an inverted terminal repeat, so the 2 ends of the displaced single strand hybridize to form
Okay, a duplex.
a duplex that looks exactly like the duplex on the
All right.
fully duplex molecule.
So that serves
Right.
serves to initiate the
Yeah.
Right.
second
Yeah.
strand
Right.
synthesis.
Huh.
Right.
So after this postdoc, then I guess you set out on your own to start a lab,
I did,
right?
Yes,
Where did you do that?
at Tufts Medical School. In the
Who hired you?
Elio?
Oh, you're kidding.
No.
Oh, he's going to love this. I do a podcast with Elio every other week called This Week in Microbiology, and I know he was chair at Tufts.
Yep.
Of course, he's now retired in San Diego, but he still keeps up.
This is Elio Schechter.
Elio
Yeah.
Schechter.
Okay.
He hired John Coffin.
Okay.
You guys must have
Yeah.
overlapped. He's still
Yeah.
there. But Elio was always talking about his old department, so I'll have to tell him that. He'll listen to this anyway. He does listen to TWiV, so he'll get a kick out of it. So he hired you. Great.
Yes, he did.
Good job,
So,
Elio.
when you go to give a job seminar, you've got a portfolio, right?
Right.
And I know what's coming up here, but what did you tell them you were going to do?
I told them I was going to continue to work on adenovirus replication.
Okay, good.
You know something that I don't know. Okay.
Well,
oh, of course.
I mean, did you continue to work on adenovirus?
I did, in fact. Yes.
Okay.
Yes.
For how long?
Until I left Tufts, actually.
Oh, okay.
After
So
4 years.
So herpes doesn't happen until down the road.
Right.
Okay.
So you were at Tufts for 4 years, working
Right.
on that, and similar aspects of adeno DNA replication.
Yeah, basically continuing on.
Okay, and then you left Tufts to
Yes.
go where?
NIH.
And there's where you took up herpes.
Right.
Why did you go there?
For family reasons.
Okay. Did you like Tufts?
I did.
Good.
Yeah.
Was it a good environment for
Yeah.
a new assistant
No, it was
professor?
great. It's a great, and as you can imagine, I mean, any department headed by Elio was a very convivial place.
Were there any virologists there that you interacted
Well, John
with?
Coffin.
John Coffin, right?
Yeah.
Okay. And at NIH, you went to the laboratory for viral diseases, right?
Right.
And who was the head of that at the time?
Bernie Moss.
And still is? Is that correct?
Still is.
Yeah. And will be indefinitely.
And he probably loved the idea that you were doing DNA
Yeah,
replication.
so I had actually known Bernie because when I worked on vaccinia DNA polymerase
Right.
as a graduate student at Hopkins, I drove down to NIH and got my stock of vaccinia virus and HeLa cells to propagate it in from Bernie's lab. So, and because I was not all that great at keeping HeLa cells going, I would go down there every few months to replenish my supply.
So there you continued initially adeno, but then switched.
I decided I needed to switch, and so I thought that moving to NIH was a good
Okay.
chance to do that. So,
Why did you want to switch?
well, because I thought that the adeno problem was basically solved at that point, or at least at the level that I wanted to work on it.
And so then you thought herpes was not, right?
Right.
So no one had purified the polymerase from herpes?
People had purified the polymerase from herpes, but there was a lot of uncertainty as to what the rest of the participants of replication were.
At that point,
At
was there a
that point.
genome sequence for herpes?
It was just being worked on.
Okay. So you really didn't even know what the genetic content was.
Right.
Okay.
Yeah, I mean there was a few DNA minus mutants of herpes that had been mapped that weren't mapped to the polymerase, so it was clear there was additional genes involved in DNA replication, but they really weren't all known.
So what was your goal with herpes? What did you want to accomplish?
Well, what I really wanted to do was do what I had done with adeno, set up an in vitro system and purify all the
Okay.
proteins and see how it worked. But so for the first 6 months I was at NIH, I spent all that time trying to make extracts from HSV infected cells
Right.
and try to just, I was hoping things would work out just the same way they did with adeno, but
Yeah, in a week.
unfortunately they didn't. So, yeah. So I had to take a different approach.
Eventually it did work out, right?
Well, I never did, and I think to this day nobody has gotten an in vitro system that actually replicates HSV DNA.
So
From beginning to
it's all
end.
done in cells.
Right.
So what was the approach? I love this.
Well, so I thought, since I couldn't get an in vitro system with extracts, the next best thing would be, or a different approach would be to systematically find out all of the HSV proteins that were necessary, and then overexpress those proteins and put them together and get an in vitro system that way. So the first thing I had to do was figure out what all the viral gene products were that participated. And so we set up a transfection assay in which we basically divided the HSV genome, which is a pretty big piece of DNA, 150 kb, and showed that you could transfect in pieces, the entire genome in pieces, and that there was enough gene expression that occurred from the transfected DNA that all the proteins necessary for replicating HSV DNA, and at the time the origin of DNA replication of HSV was known. So the assay was to ask for the replication of a plasmid containing the origin of replication. And so once we showed that you could do that with fragmented DNA, then it was just a matter of grinding through and cloning and whittling it down to the unique set of genes. And fortunately, at that point, the HSV1 sequence, DNA sequence came out, so that made life a lot easier.
So then you knew kind of where the start and stop for individual open reading frames were. So you had a candidate open reading frame, made it easier to clone that open reading frame. So actually something just occurred to me about this, and that is that during a normal herpes virus infection cycle, there's immediate early gene expression that then activates early gene expression, and the DNA replication proteins are early genes. But in your transfection assay, ultimately, you don't have that first step of activation by immediate early genes necessarily. Ultimately, once you've whittled
well,
it
actually,
down
we do, because in the transfection DNA replication assay, you needed to include the immediate early genes to activate the early genes.
Okay.
But that paradigm was already known. So.
Okay. So that those immediate early genes just kind of become background in the assay.
Right.
So then ultimately you must have cloned all these downstream from some other promoter or something like that? Did you go?
Right, did that.
All right.
So how many gene products did you need to replicate a herpes origin containing plasmid?
7.
Okay, and that's this paper here in 1988. Wu, Nelson, McGeoch, and Challberg. Duncan McGeoch, was he a postdoc of yours? Or?
No, he was at the Institute of Virology in Glasgow. He did the DNA sequence. So he made DNA sequence available before it was published.
So people are going to cringe that I called him your postdoc, but that's okay. It says seven proteins. I remember this paper. I was just 6 years at Columbia, and I remember seeing this and people talking about it in journal clubs. What were the 7 proteins? You knew at the time because the sequence had been done. McGeoch.
Right. So the 7 proteins were 2 subunits of the DNA polymerase. So there's a catalytic subunit and the processivity subunit for DNA polymerase. 3 subunits of a helicase primase complex. A single-stranded DNA binding protein, and an origin binding protein.
Wow, that's beautiful, isn't it?
Yeah.
Well, I remember this as a tour de force.
Yeah.
Of course, in my conception, it happened overnight, right? Because I was just sitting around having a good time, and then all of a sudden, this paper comes out that's got all of this stuff in it. But it really it's a novel approach to identifying these things, and it gives all of this information in one big dump. It was amazing. It's really great.
This starts a new field because then everybody else starts to look at individual proteins, and they could alter them and look at the activities in your system, right?
Right.
I presume you did that as well, right?
Right.
So what are the...
I was actually more interested. I mean, we did some of that, but I was more interested in the enzymology.
Mm-hmm.
So how long did that take you from getting the idea of the transfection assay?
It was amazingly fast. I mean, it was under a year.
Okay.
So was that mainly the first author here, Wu?
She was my first postdoc.
Okay.
But I'd actually...
You worked on it as well yourself.
I'd done a lot of it.
Yeah.
It went way quicker when we got the DNA sequence, because
Yes.
what I was doing for a while was to take it down to a small restriction fragment
Sure,
and then do
sure.
exonuclease, digestions of either. That was kind of tedious.
What this makes me think of, which I'm sure you'll appreciate as a biochemist, is that it's all in the assay, right?
Right.
If you have a really good, solid assay to do something that's reliable and reproducible, once you've got that, things can move really fast.
Hershey Heaven. Remember?
Hershey
Yeah,
Heaven?
absolutely. Hershey Heaven.
Have one good assay.
this
One
is
good
a-
assay and just do the
okay.
crank. Yeah.
So you had an origin-containing fragment cloned on a plasmid, and you put this into cells. So this was this a stable cell line that had this plasmid, or would you put it in each time?
No, it was a part of the transfection.
Okay.
And then the assay makes use of this restriction enzyme anomaly, the DPN1.
DPN1, okay.
Yeah.
Why don't you explain that, because the listeners may not know it, even the virology listeners may not know it.
So, boy, it's been such a long time. How does this even...
So a plasmid grown in E. coli is methylated by the damn
Methylate,
methylase,
right.
and that makes it resistant to, sensitive to DPN1, a restriction
Right.
enzyme.
Right,
But if it's replicated in eukaryotic cells,
right.
you don't get that methylation. So you look for DPN resistance of the replicated plasmid.
You extract plasmid DNA. You probably did a Hertz supernatant, right? That's how we...
Yeah.
So,
Actually, you can't do quite a hurt because HSV DNA gets replicated by some kind of rolling circle mechanism,
it's
so it
big.
ends up quite big.
Okay, yeah. So you just extract DNA and cut it with DPN1 and look
Right.
for something that can be cut, and
Right.
that's been replicated. Beautiful. Many people use that assay.
Yeah.
Right. That's great. What other things? How many years did you spend at NIH in your lab?
17.
Okay.
Wow.
What
What other things did you work on? Mostly herpes replication?
It was exclusively herpes
Okay.
replication, yeah. So, as I said, I was mainly interested in the enzymology and what these, because when we defined these genes by transfection, we didn't know what most of them did.
Sure.
So the first thing was to try to figure out what they did by overexpressing them. So each protein was overexpressed in baculovirus expression systems and assayed for various activities relevant for DNA replication.
I seem to remember you also defining the number of origins in the genome. Was that your work?
No.
No? There's more than one, isn't there?
There's 3, actually.
Okay. So did other people buy into this system and start doing work on it?
Absolutely, yeah.
And to this day, do people still study simplex DNA replication?
Yes. And as I said, it's proved to be, for reasons that just aren't clear, an intractable problem to get it to take these 7 proteins. You can purify them and demonstrate that they have enzymatic activity, and you put them together, and with an origin-containing plasmid, nothing happens.
In vitro, you mean?
Right.
Ah, so that was your original goal, right?
Right.
Still hasn't been achieved.
Still hasn't been achieved.
But you don't know why it doesn't work,
No.
right?
Do you know that those are actually the origins that are used in cells? He's thinking, by the way.
It's
Yes.
okay. We don't mind. We don't mind pauses.
That's all right.
Well, what would be proof that was the case?
I don't know. I mean, can you like mutagenize them? And well, I don't know how you would do that because you'd wind up with something that's dead, right? In cis.
Right. I mean, you can only ask those questions not in the intact genome, but in a transfected plasmid. I guess the best evidence, well, not the best, the way these sequences were originally discovered was if you passage herpes virus at high multiplicity in tissue culture, you end up getting defective genomes, and the defective genomes turn out to be repeats of a sequence which is the origin of replication. So
it turns out, right, that there's a lot of organization. I mean, when herpes infects a cell, it sets up these replication centers in the nucleus. There are host proteins that are recruited to these. So it strikes me, thinking about this, that it could be that a lot of what's missing here is host proteins and some sort of larger structural components, scaffolding of one
right.
sort or
Chaperones,
another, chaperones, and this kind of stuff. Right?
Yeah.
Do you keep up with this field now?
A little bit. I mean, now that for when I started in extramural as a program officer, I didn't have any responsibility for herpes viruses at all. It was all pox viruses. And so I didn't really keep up that well, but I've recently gotten back or gotten part of the herpes virus portfolio.
So let's, if we're ready, do that transition.
Yeah, so you after 17 years, you decided you didn't want to have a lab anymore?
Yeah, I decided that I really needed to do something different. If I wanted to stay in the lab, I needed to do something very different than herpes replication, because it just wasn't, I don't know, it seemed like the questions that I was asking in my lab just weren't that interesting to me anymore. So I needed to do something different. But then I decided to do something very different and just get out of the lab. And,
so what year is this?
let's see, it would have been 2001.
So 11 years ago.
Yeah.
Okay.
I remember seeing you at a study section. I said, "What are you doing here?"
Yeah, that was not an uncommon.
And he said, "Well, I decided to make a big change."
Yeah.
I remember this. I don't know if you remember. Probably everybody said that to you, but I distinctly remember. That was my first tour on virology study section. You had said that. So you decided to become a program officer? Is that what you came right out of your lab?
Right.
Did you say, "Oh, I want to be a program officer"?
Well, I didn't even know what a program officer did, really.
Yeah, because you've been at NIH.
Right.
Right.
When I started the job, I really didn't know what a program officer did. But, I knew it had something to do with administering grants and so forth.
So did you, I mean, in your mind, it was sort of like, I want to stay geographically where I am. I'd like to maybe try out some science administration.
Right.
Oh, here's this job, I'll try that.
Exactly.
Okay.
And it worked out because it was 2001, 2002, so the whole biodefense thing had exploded.
As it were.
And they needed somebody to run the poxvirus program because nobody in the DMID extramural knew much about poxviruses. Of course, I didn't really work on poxviruses, but I was in Bernie Moss's lab, which gave me a certain amount of credibility for poxviruses.
Right. So you became a program officer. So tell us what a program officer does, because probably most listeners don't know.
Yeah, and this is key to the whole thing.
Yeah.
Right. So I think program officers have maybe three interrelated jobs that are, as I say, interrelated but different. The first is what you might call portfolio management. We administer a certain area of science, a program. In my case, it's pox viruses and alpha herpes viruses.
These are extramural, right?
Right.
That is stuff NIH funds on the outside of NIH.
Right.
Because when you worked in your lab, you were an intramural program, right? Okay.
Right. Which do not, I mean, as Rich alluded to, people who work in NIH intramural know nothing about NIH extramural, because people who work in NIH intramural get their money from a completely different mechanism. They don't have to apply for grants.
So extramural is where we started with this conversation.
That's
People like you and I, Vincent, submitting grant applications.
Right.
Those are extramural grants, and program officers deal with that stuff.
So you have a portfolio of grants. So initially you started with pox viruses.
Right.
And if grant applications would come in, they would be part of your portfolio.
Right.
So what does that actually mean? And you just oversee them?
Well, so I guess the core job of a program officer is to oversee these grants and make sure that the investigators comply with all the relevant laws and policies regarding the use of grant money. And so for example, before somebody before their grant gets paid, I have to go through a checklist of things that ask various questions. Does this person comply with the policies regarding human subjects?
Mm-hmm.
Is the animal work all okay? Is it approved by the Cook and their facility? Does it involve select agents? All these sort of things that are a little bit too scientific for an accountant to be able to figure out, but on the other hand, are not that difficult.
Right. So when we are at study section, program officers usually sit in the room.
Right.
So you were there today. What are
Right.
you doing while you're
Well,
sitting?
okay, so one of the other big jobs of program officers is to deal with various peer review issues. So once the review is over, then the SRO and the people who run the review don't really have anything to do with the applicant. So because funding decisions for individual grants are not made by the review people, they're made by institutes. So
we are told that all the time,
right.
very explicitly. You are
Right.
not making funding decisions. You're evaluating the scientific merit of these applications, and they get passed on to the people who actually hold the purse strings.
Right.
To make the funding, which is you.
Which is right.
Yeah.
Well, program officers don't really make the funding decisions,
Right.
but
Okay.
the institutes do. But so why do I go to study sections? One is that after the study section takes place and the reviews are out, then I get a lot of calls from applicants whose grant was reviewed wanting to know what happened, why their grant didn't get a good score, basically. And they're looking for some extra insight that's not conveyed in the summary statement. And I can't usually give them any additional insight, but sometimes I'll say, well, there's all this stuff in the summary statement, but the thing that really drove the score was this one issue, and that's the issue that you need to take care of in your revision.
One of the things that I think happens, and I'd be interested in your insights to this, is that lots of times grants come through, and the problem is that they're boring, right?
Right.
But the reviewers don't know how to say, this is boring. And so they pick a bunch of stuff
Right.
to say
Right.
to justify the fact that they're giving this a lousy score,
Right.
rather than saying, this is boring.
And one of the difficult jobs that I have is I try to, and I'm not very good at saying this gently, I think, but I try to give people an honest assessment of what what really went on. And I've told many people, look, you can read the summary statement and read those criticisms all you want, but that's not the main issue. The main issue is that they're just not excited by this.
Right.
And they're never going to be excited.
Right. And that's really an important job, to communicate that.
Yeah.
Yeah, because it's not written down.
No,
Right.
That's
it's not
really...
written.
yeah.
Well, as a reviewer, if that's my problem, I try and write that down as nicely as I can so that they get the idea. But I don't know how common that is.
Yeah, and these sub-scores have actually not helped that whole problem, because especially new investigators, new applicants, they go through and they read the criterion scores, and they say, they really thought that investigator was great, and my institution is great. And I said, don't read anything in that summary statement that's positive. Just ignore it, because they're just throwing you some bones. You need to focus on the negatives here, because that's what you need to change. And it was only the approach.
Only.
Only the approach.
Only. So in the old
Yeah.
days, you just gave one score to a proposal, and you're referring to the fact that now we have subcategories, which
Right.
include not just the approach, but investigator and environment.
Right.
Quite often, most of them are similar for most grant applications, right? It's really the approach and then maybe significance
Yeah.
is what's really differentiating. So I interrupted you. You said you had 3 jobs as a program officer, and the one is to deal with the administrative science that can't
Right.
be dealt
The other
with.
is to deal with peer review issues, and another aspect of that is, and this varies from institute to institute, but NIAID mostly funds applications strictly on the basis of score. So we establish a pay line. If your score is better than the pay line, you get funded. If it's worse than the pay line, you don't get funded. But we do make some discretionary awards, and that's based on program officer recommendations. So we try to, if we think there's a area of research that really needs to be funded, and the applicant, and we're going to lose this investigator because he's going to have to dismantle his lab if he doesn't get a little extra money, we'll make a discretionary award. And
So that's special pay.
That's special pay, select pay,
Select
bridges, that kind of...
It's probably the most interesting part of the job, right?
Right, exactly. Although, there's not, in NIAID, as I say, there's not that much money for discretionary awards, so it's very competitive.
Has it changed over time? Did it used to be more?
No.
But this depends on your scientific judgment. So
Right.
it's based on your experience, which is good because
Right.
you have that experience. And that's why you get assigned certain viruses, right?
Right.
And you now have pox and herpes, alpha herpes.
Alpha herpes,
Not all herpes, just
Right.
alpha.
And, the herpes is a big field.
Right. So you have people to do the other herpes viruses,
Right.
program officers. Okay.
Right.
So there's another function that I think of for program officers, and that is that as investigators, we're often encouraged when we're considering writing a grant application to contact the appropriate program officer and say, I've got this idea, I'm thinking of writing a grant, am I wasting my time or whatever, and get some spin from the program officer. I'd be interested in your perspective on that.
Yeah, I don't really find that I have much to say to people who ask me that.
Okay.
Because if just the way we're set up is there's so little discretionary money. If an application comes and gets a good score, if it's assigned to NIAID and it's assigned to my program and it gets a score below the pay line, we pay it. So it's, it's pretty cut and dry. But there is one other thing that we do that somewhat relates to that, and that's dealing with the whole area of initiatives. So program officers are sort of the front line in the development of funding initiatives, and that's really special pots of money that I kind of think of it as jump-starting a certain area of research. So if we think that there's something lacking, there's an area of research there's just not enough people working on that really needs to be pushed, then we'll say, well, we need an initiative in this area, and there's this whole process for doing that. But if it's approved by the upper management at NIAID, then we write it up. That goes out into the community, and it says we're looking. So a typical initiative would be what's called an RFA, a request for applications. So we're looking for applications in this certain area, and we're setting aside a certain amount of money that is devoted to that area. So please send us those applications.
So when you say we, administratively, is this like a gaggle of program officers in NIAID or what?
Well, I think mostly when I said we there, I was referring to NIAID. But as I say, initiatives are started by program officers.
Okay.
So
if you had an idea, you could start it.
Right.
It doesn't have to originate in a meeting with a bunch of people, right?
Okay.
And we, as I say, we have this, NIAID has this whole big process. So in the Division of Microbiology and Infectious Disease, program officers bring initiatives forward. They're talked about, chewed over, and then they kind of move up the chain of command. Eventually, they'd be signed off by somebody by the director. Then it goes to council. The council has to approve any initiative concepts that are brought up. And then if council approves it, then it comes back to the program officer whose idea it was initially to actually write the initiative, to write the RFA,
implement the whole thing.
But it all starts with program officers, and that's really the third area that I think program officers do, is they're kind of advocates for a particular area of science. For pox viruses and herpes viruses, I'm the person in NIAID who has to say, this is important. We should be doing more of this.
And here is where I am really grateful for your 20, 30 years of genuine academic science background, because I know that there's a guy at the helm at NIH who knows what it's like and knows what's going on, who's helping make these decisions and guide things.
In the interest of full disclosure, Mark has been your program person,
yes,
right?
That's right.
Yes.
For many
program
years.
officers since 2001.
Right.
Since he was starting, yeah. So do you mainly deal with the investigator-initiated proposals, or do you do program projects, grants, all those? You do everything.
Do everything.
Anything within NIAID you could.
Right.
And then, of course, each institute has its own program officers, and they're all separate from you as well. Is there any crosstalk between institutes?
Very little between institutes, yeah. I mean, as you probably know, many grants could potentially be of interest to more than one institute, and so they get assigned, doubly assigned, dual assignments. in practice, that really doesn't
Yeah.
make much difference.
So how many program officers are in NIAID?
I think there's in the neighborhood of 50.
Okay.
I know mine,
Yeah.
Other people, other viruses, they have their own officers.
And this is the National Institutes of Health. How many institutes are there?
And there's more than 10,
Okay.
11, I think.
So that adds up to a fair number of program officers. Okay.
So in these years that you've been, over 10 years you've been doing this, have you seen any trends in the way science is done in this country?
Well, clearly over time, science has been getting bigger. The DNA, the genome, the omics efforts, whatever it is. There's certainly a lot more of that kind of collaborative thing, or multi-PI operations, where I would say that's the biggest change over time.
Does NIH like that? Does NIH prefer that to little grants?
I'm not sure we prefer it, but we certainly go with the flow on that.
So if people, you do have mechanisms that
Right.
allow these multi-university grants. So if it's good, you will go with it.
Right.
So here's something that probably young people in the field would like to hear about, right? It's no secret that it's getting tougher and tougher to get an NIH grant. And it's probably going to continue to get tougher. So what do you tell people who are just coming into science? Should they not come into science? We don't want to dismantle our wonderful thing here.
It's very tough. It's hard to know what to tell people. I tell people, unless you have to be doing science, you probably shouldn't do it. There has to be an overwhelming desire on your part to do it, or it just is too hard. And it's still hard. Things are getting tougher and tougher, and there's programs that we have, that all the institutes have, to make things a little better for new investigators. But in many ways, I'm a little concerned about those, because we're kind of delaying the hard part. We're pushing off the hard part until the first renewal, or when they're no longer a new investigator. So I don't think we can it's good to give new investigators a little bit of extra help, but too much is doing them a disservice, I think. Is it better to decide after your postdoc that you need to do something else, or wait until you can't get your first grant renewed 5 years later?
By doing science, really is not necessarily don't do science at all, but don't try and do academic science.
Academic science. Don't be a PI in
right.
academic.
This reminds me of the conversation we had with David Baltimore, who said, this is very difficult. And it's the same advice that my daughters got when they were thinking of going into theater, okay? And the advice there is, if you can imagine yourself being happy doing anything else, do that.
It.
Because this is hard.
Yeah.
But if you can't be happy doing anything else, if this is what you have to do it. It makes sense, because that's the kind of passion that's required to make it work.
Absolutely. And, things sound, it's certainly way harder to make a career as an academic scientist now than when I was starting out. When I put in my first grant application, I fully expected to get it funded, and it was. And, it just wasn't that hard. And so I think it takes more work now. I think new investigators have got to have some expectation that their first application is not going to succeed. But, I think for people who are persistent and keep after it, it's still possible to make a career.
Mm-hmm. So when I came into science, when I started my own lab in '82, you had about a 30% chance of getting your first grant, which was pretty good.
Right.
The pay line was about in the 30s.
Right.
It's slowly gotten lower and lower. So why is that? Is that because science is expanding, more people are doing it, and the money is the same? Or I don't understand why we're in this position.
Well, yeah, it's sort of out of my... I certainly don't have a complete grasp of why it's gotten quite so bad, but clearly there's a lot more people doing it now than used to. Because if you look at the amount of money that goes into investigator-initiated grants over time, it's gone steadily up. There was the doubling, back in the 90s. But what the doubling did was to convince a lot of academic institutions that they needed to expand their faculty, and so with the doubling of the NIH budget, we got a big increase in the number of applications and investigators. And then when funding leveled off, the pay lines dropped precipitously because we'd made commitments, out-year commitments to all these new awardees. So, grants are for 5 years. So, and we have no idea what the budget is going to be 5 years from now. So it makes life very difficult for the financial planners.
I think the universities need to commit more money to supporting basic science. As you said, when it used to be a university would put up a new building and assume they could fill it with people who had R01s.
Right.
And as everybody knows, when you get a grant, you also get money, indirect costs for the institution, and that's how universities have thrived. But they have to change that way of doing business. It doesn't work anymore. If universities could contribute a substantial salary for all their faculty, this would free up research money. I don't know if that's a naive view, but that's what I've always
said. I would agree with that. I think the universities have become enormously reliant on NIH to kind of help the universities do their thing, and that just can't go on forever.
But I think universities would say, well, if you're going to ask us to do that, what's our mission and how do we make money? We teach students and we get, we pay for that by tuition. So if we're going to pay for you faculty, we're not going to be able to have as many as we do, and you're not going to be able to spend your time doing research. You're going to have to spend more of your time teaching.
Right.
So,
a great university is great because it has a research faculty, right? Who are doing stuff, no matter what field it is, but let's pick science. They're doing research supported by NIH. That's what makes them a good faculty. They can teach right from the heart, right? So it behooves the university to have those kind of people doing research. You can get support from NIH, but also contribute to them. Now, the money situation is difficult. Tuition won't pay for all the professors at the big universities. It's a problem. I don't know how to solve it, that's for sure. Do you know, Rich?
No, unfortunately, I'm not in a position of having to come up with that answer.
Yeah. But I think going forward, the people who are getting in the field have to think about this and figure out ways to get around it. Because you and I are reaching the ends of our careers, right? At least research careers.
Yep.
And but I think I would love to contribute something, but I don't know how to solve it. I'm not in any position to do that.
Well, we can ask the questions and broadcast them.
Yeah.
Maybe stimulate some discussion.
But I'm sure you agree that doing research is essential for the world the way it is now, right?
Absolutely.
So how do you like being a program officer?
I like it a lot. Yeah, it's different than going into the lab every day.
What do you like about it?
I like that I get a much more global view of at least one area of science. You know, it's the bird's eye view rather than the worm's eye view. And I do a lot of different things.
Yeah, you've been involved in more than just the activities that you've talked about, right? What other sort of things do you do?
Well, NIH also, in some areas, for example, and particularly in the biodefense area, actually has contracts to develop products. So, you know, the area I'm in, for example, is smallpox, new smallpox vaccines and drugs, anti-smallpox drugs. So, since there is no smallpox in the world right now, we're only looking at a possible terrorist scenario, or the only customer for smallpox drugs potentially is the government. So we can't really ask pharmaceutical companies to develop a drug on their own dime and then have it sitting around in the event that we might need it someday. So basically, NIH is in the position in that sort of arena where we have to be the driving force for developing new vaccines and drugs and other therapeutics and so forth. So I'm involved in a lot of that, which is also interesting.
Do you work with BARDA?
Yeah.
Work with BARDA.
Oh, so you must know Ed Niles, right?
Yeah, I do know Ed.
So it's perfectly appropriate that we're in his house.
That's right. Absolutely.
Thanks, Ed.
In fact, Ed and I worked on same projects.
And, well, we talked with Ed a year ago, maybe?
I can look it up. It's the Ed Niles, a KM VMAX kind of guy.
Yeah.
I'll look that up.
That's what we called Ed, a KM VMAX kind of guy.
Right.
What would you call yourself?
That's how he described himself, right?
You're not
yeah,
a,
what would
you're
you
not a
call
KM
yourself?
VMAX kind of guy.
Not really, no. I don't know what I would call myself.
You have to come up
A
with
man
title.
for all seasons.
I mean, in my heart of hearts, I think I'm still a cold room kind of guy.
A cold room kind. Oh, that's great.
Excellent. A cold room kind of guy. Excellent.
You didn't smoke, did you?
No.
I knew a guy who was a big biochemist, and he used to do columns in the cold room, and he'd stand outside the cold room smoking a cigarette, watching it drip, and then periodically run in and do things, and he didn't want to stand inside and get cold. Cold room kind of guy. So you had left the bench because you wanted to do something totally different. Are you going to want to do that again, or is this the...
I think this is the end.
Okay.
TWiV 120, February 13th, 2011. Ed Niles, a KM VMAX kind of guy. So, Vincent, that was over a year and a half ago.
Yeah. That's the last time we were here in Alexandria, I guess.
Mm-hmm.
Right? Well, I think that's a good story, don't you, Rich?
Outstanding. I've always thought, well, first of all, I've always had a lot of respect for your science, Mark.
Thank you, Rich.
And I've always appreciated you being there as a program officer. And so I'm happy to have this opportunity to kind of tie all that together and tell the rest of the world about it. And I think it's also important. I mean, we can provide insight to people as to how this whole process works and give some inside insight as to, what goes on. And I appreciate the opportunity to do that.
Yeah, we never had anyone in program person on TWiV, of course.
No, I don't think so.
I think you're unique because you made this switch. And if, would you say to young people or mid-career people that are thinking about it that would be a good thing for them to do?
Yeah, I do. It's way different than doing science, but it gives you certainly a different perspective on science. And I would say, to follow up on what you said, back when I had a grant and was, a young PI, I had really no clue what went on in NIH program or what the whole, I just wrote an application and sent it in, and a while later they sent me some money, or not. But I think it's important, really, to understand. It's gotten so difficult that it's actually behooves investigators to understand the process.
Well, actually,
because it makes a difference.
When I was first starting out, from my perspective, the administrative side, the program officers, that kind of side of NIH was always really scary to me. And I didn't see them as advocates, actually. Okay, and now I realize that I was completely wrong.
Right.
And that in fact the program officers and the administrators at NIH are very much advocates. You guys want to see the science advance and want to see it advance in the right way. And that's an important thing to communicate. Actually, I have another question. You have a very robust scientific background going into this. Is that common for program officers?
I would say mine is a little more unusual, but most program officers have had some experience as investigators on their own.
Okay.
So we don't take that many people as program officers, say, straight out of a postdoc. They're looking for a little bit more experience than that.
That's good, because I think it's reassuring to know that the people who are on the administrative end of this know what it's like to be in the trenches.
Yeah.
I can't resist one more question. So if you were going to go back to the lab today, right? You found the fountain of youth, but today, what would you work on? Anything in the not just viruses if you want, but I'm sure that's what you know best.
I think it would be something much more involved with closer to the disease potential of a virus than the basic science, I think. Because I've just gotten more aware of the public health implications of different viruses. It would be something like dengue, something like that.
Pathogenesis.
Yeah.
Okay. So we do a couple of emails.
Yeah.
I have some follow-up from last time. The first one's from Suzanne. Your most recent TWiV in Nebraska made me want to add a few verses to that old kid's song. There's a virus in the alga in the paramecium, on the speck, on the flea, on the wart, on the frog, on the bump on the log, in the hole in the bottom of the sea. Just thinking, and thought it might amuse you guys too. So we did the last TWiV in Nebraska, and Jim Van Etten was telling us about these algal viruses that he works on. They infect algae, and the algae are inside of paramecia, and that's where that came from. The cool thing is, my understanding is when the alga is in the paramecium, it doesn't get infected. Apparently the virus is compartmentalized in the paramecium. It doesn't infect the alga, and then something disturbs it and it gets infected. We never got into it. We have to have him back to talk about that.
Yes, I have to review this every time I talk to Jim because it's very confusing.
We'll get him on to explain. He said he'd come back and do it. Jim writes, with respect to TWiV 200 video, that's the needle video, will we finally see a naked Racaniello?
I don't think so.
Well, he's going to have to wait and find out.
When's that coming out? Do you know?
I don't know. A few weeks, I guess. I think it's halfway edited. It takes a long time for them to edit it. Next one's from Claudio. Hello, congratulations on surpassing 200 episodes. I've been a rabid fan of TWiV, TWIP, and TWIM since I first stumbled on TWIP. I look forward to every new episode. They've helped me get through my days as a small electronics technician and have made multiple road trips bearable. This series of podcasts awoke a latent interest in microbiology. Time to make something for TWiV. I've attached a picture of some simple plaques I've printed on my do-it-yourself 3D printer. And he made a plaque of a bacteriophage, and next to it it says TWiV 200.
This is a serious pun, you realize. Those are plaques.
Ah, you're right. It is a serious pun. That's excellent.
They look cool.
They're really cool. Thanks for all the hard work and time you folks put into these podcasts. I look forward to more. Let me know where to send these plaques, and I'll send them on their way. So I'll let you know where to send them, Claudio, and I'll distribute them to you guys, because Rich and Alan
Oh man, I would love to have one of those.
should get one. Yeah.
That'd be great.
The next one is from John. I've been working on a transcript for episode 131, a rheostat for cancer. I hope I'm not duplicating anyone else's efforts. So if anyone was thinking about it, don't do that one. John's transcribing that one, which we really appreciate. Thanks for the wonderful podcast. I listen to them while I do all manner of stupid things like cooking, cleaning, etc. I must admit my apartment got a lot cleaner after I found TWiV and TWIP. And John is a postdoc in mathematical physics. How about that? I love the people that we get.
Yeah.
Hey, Rich, can you read the next one?
Sure. Catherine writes, this may be a bit random, but here it goes. A friend of mine, a virologist, absolutely adores your podcast. She's done me a great favor, and I'd love to say thanks with a little gift. I was thinking of a magazine subscription, but I'm not sure what to go for. I want something on the geekiness level of TWiV, so a bit more geeky than National Geographic or New Scientist, that is affordable for a normal person. She is relocating to the States this week, so maybe you guys have some magazines that would be perfect, but that I'm not aware of. Do you have any suggestions for me? Many thanks and kind regards, Catherine. Vincent, you have some suggestions.
Well, I just thought of some science magazines, but I don't know if they're geeky enough. You know, Wired, Scientific American, Discover Magazine, Science Magazine. If she's a scientist, she may get that already. There's American Scientist, Seed, Science News, Popular Science. I've sort of seen every one of those. Those would all be good, probably. Do you know any others, Rich?
No, I think that pretty much covers it. Your first two here, Wired and Scientific American, I think are really good really ideas. And they come, my sense is that they're kind of different perspectives.
They are,
So,
yeah.
Same thing.
I mean, of course, Scientific American has articles mainly by scientists.
Right.
Right, and so it's different from Wired, which hires professional science writers, so you could take your pick. They both have online versions.
And tell me if I'm wrong, but it seems to me Wired is kind of hip science.
It is.
And Scientific American is the old stuffy scientist kind of science, all right?
I think, I'm sure Scientific American would love to change that.
Yeah, it's cool though. I mean, Scientific American has a lot of history to it. It's a great magazine.
You know, we just got a subscription for our kids. I haven't looked at it in years.
What, Scientific American?
Yeah. So years ago I used to look at it, but I haven't in years, and I looked at it and I actually enjoy it. It's good. It's good to see what's going on in other areas of science in particular.
Yeah, my father subscribed to Scientific American, and I used to look at that. It's got very, very good, very readable,
yeah,
but accurate scientific articles. I think probably more in-depth and stuff than you would find with
sure,
Wired.
absolutely.
And a lot of features like history of science and mathematical problems and that kind of stuff.
So the latest issue I got, I opened it up, and on page 3 was a full-page ad by a bio supplier company, okay, that you would know. It says, the NIH budget may be cut $2.5 billion. Here's why it shouldn't happen. It gave a whole list of things, why this can't happen, you know, shut down productive labs, put scientists out of business, interrupt productivity, you know, go to the website for more info. I thought that was pretty cool.
Mm-hmm.
Right. $2.5 billion would be a big deal, right?
Well, it's what, 8%?
Yeah. Wow, that would be a problem. Should we do one more, Rich?
Sure.
Let you take that next one, too.
Dear TWiVers, I am a fourth-year PhD student at Yale University in the Environmental Engineering Department, working in an environmental microbiology lab. The weather in New Haven these days is scorching, but is scorching hot with the occasional summer thunderstorm. A great deal for my advisor, who can be assured that I spend the majority of my time inside our building, as my home lacks air conditioning. I'd like to comment on your amazing podcasts. I'm a regular listener to TWiV and TWiM. I especially enjoy your coverage of the human microbiome. Get Joe Handelsman on TWiM more often, and the talks about influenza transmission. My PhD work centers on the influence of the human occupant on the indoor air microbiome. And thus these topics are dear to my heart. In TWiV 191, you talked about influenza transmission via the airborne route, droplets, and contact. The question about virus inactivation in air was raised and remained unanswered at that point. I'd like to take this opportunity and hint to a great modeling work by Lindsay Marr of Virginia Tech that combines dynamics of influenza virus containing aerosols influenced by temperature, humidity, as well as varying air exchange rates in the room, with virus inactivation parameters to estimate removal rates from air. Her results indicate that virus inactivation is important for virus removal from air and largely dependent on humidity. Virus inactivation and humidity are positively correlated. The modeled air exchange rate, AER parameters, 1 or 10 in the paper, are very reasonable for mechanically ventilated building as often found in the U.S. But having some experience in measuring air exchange rates in public buildings and schools, one air exchange rate per hour sometimes is, sadly, high in those common spaces and may partially explain why we catch a cold in places such as classrooms and waiting rooms. Removal by deposition, air exchange, and inactivation should be kept in mind when the infectivity of influenza is discussed, especially under the recent focus by the academic community. As Lindsay shows the importance of humidity on virus inactivation, she had also mentioned, not sure if that is listed in this paper, that protein content in saliva influences survival rates of viruses. High and low content are good for survival. Did you know that infants and adults have different saliva compositions with regards to protein and salt content? Isn't that mighty cool? So again, another parameter is added. In summary, one has to look on a much broader scale and might have to take many more factors into account than previously shown when accurately estimating disease transmission via the airborne route. The paper is published in PLOS One and thus accessible to all listeners, and she gives a link. Great podcast. Keep up the viral work, Nina. And she's, as she said, a PhD candidate at Yale in environmental engineering. Well, that's cool.
Yeah, so basically, we knew that humidity was bad from the Palese work on transmission in hamsters. So that's why flu probably predominates in the winter when there's low humidity. Mm-hmm. I didn't know this about the air exchange.
Right.
You know, we're always complaining in rooms where the ventilation rate is high, but it's probably good for us, especially when, like in a study section, when there are a lot of people in a room.
Right. A lot of virologists.
Yeah.
Right.
And the saliva business is pretty cool. I didn't know that. All right. Well, thanks for that. Let's do a couple of picks. What have you got for us today, Rich?
Got to get down to the bottom here.
Yeah, long list of email.
Okay. I've been wanting to do this for a while. I've been conscious of this. Almost there. Right. Mattel Toys has a new Hot Wheels. And it's the Curiosity Rover.
This is great.
I'm not sure it's actually for sale yet. I've seen a couple of articles about how this is going to happen. Isn't that
way cool?
I've seen a couple of articles about how this is going to happen, and I've looked on the Mattel website and stuff, and I haven't actually seen it, so I'm not sure it's in stores yet. But, you know, keep a lookout for this. I have to get one of these.
These are the new ones. The new one that just went up. Is that right? It says, 2012.
Yeah,
wow.
absolutely.
Cool.
So this is the new Mars rover, and it's being made by Mattel, or is going to be, as a Hot Wheels car, which I think is just great. All right, to have
Very
somebody
cool.
like Mattel selling state-of-the-art, up-to-date science toys for kids.
Carries the tools such as a
I
drill.
want to see my grandsons running Curiosity rovers all over their living rooms. Matter of fact, I ought to buy a few of these and give them out.
Carries a drill camera and a laser to see if Mars could have ever supported small life forms called microbes. You know, if there are microbes, there are viruses.
You bet.
It's pretty neat. My pick is a special issue of MBio, which just came out. And it has a series of articles debating should the moratorium on research enhancing H5N1 transmissibility end. Has articles by Arturo Casadeval, Ron Fouchier, Tony Fauci, head of your ultimate boss, right?
Right.
Mark. Mark Lipsitch and Barry Bloom, Ian Lipkin, and Stanley Falco.
Wow.
All
That's quite a lineup.
with their own viewpoints on should we allow this to go forward, should we raise the moratorium or what. So I think this is pretty cool for everybody. We might even want to discuss this at some point on TWiV, so here you go, you can check it out.
Do your homework.
I'm linking to a blog post on MBio that links to all the articles, so it's easier to find them. I think that's pretty important stuff, especially since we talked about it so much here on TWiV. And we have a listener pick of the week. This is from Claudio, who had made those nice plaques for us. I'd like to offer up a couple of related readers' picks for you folks. I came across this article in Ars Technica entitled Do-It-Yourself Lab Equipment, courtesy of 3D Printing. 3D printing has been around for some time now, mainly in larger industry and academic institutions. The hobbyist and do-it-yourself community has been working hard on bringing these kinds of tools to those willing to do some work. My open source 3D printer is a Prusa RepRap, similar to the one linked in the article. I was lucky enough to be a member of the local hackerspace makerspace called Mid-South Makers, and with the help of a fellow member with a MakerBot, we were able to print the parts to put several together for ourselves. To better describe it, it's a solution looking for a problem. The open source 3D printing community extends to designs as well. There are several online repositories that host user-created designs, including many models that can help flesh out a home lab. There are Dremel-powered centrifuges, an open-source orbital shaker, PCR machine replacement parts, and even a portable cell lysis device for DNA extraction. And for a second reader's pick, I'd recommend doityourselfbio.org. That's DIYbio.org. The do-it-yourself hackerspace movement has been breaking into microbiology. DIYbio.org is an organization dedicated to making biology an accessible pursuit for citizen scientists, amateur biologists, and biological engineers who value openness and safety. Hopefully some of your listeners would be willing to help further amateur science. It's pretty cool stuff.
Yeah, I've seen this. I think we had a video on TWiV some time ago from some guy who was using this microcentrifuge.
Yes.
Where he made the little head for his Dremel tool on a PCR printer.
That's right.
And it worked just fine.
Yeah, these 3D printers are great. I know that structural biologists use them now a lot to make models. You can print viruses out in 3D and proteins, anything. It's very cool. All right, thanks for that, Claudio. And that will do it for TWiV 203. You'll be able to find this as usual on iTunes at twiv.tv. It's the Postman. Is it Journal of Biological Chemistry?
No, it's a Netflix.
Last time we were here at Ed's house, we got Journal of Biological Chemistry dropping through the mail post as we were recording. How appropriate was that? Since you're here, Ed, we want to thank you for letting us use your house.
I'm happy to have you.
Thanks very much. I also want to before I do that, let me remind everyone to send your questions and comments to TWiV at twiv.tv. Mark Challberg from NIAID. Thank you for joining us today.
Thank you for having me, Vince.
I hope you liked it.
I did.
You going to listen to this one?
What's that?
You going to listen to this one?
Oh, well, I don't know about that.
You might not want to listen to it. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Sure enough, great fun.
I understand you're going to hang out here with Ed Niles for the weekend.
Yep.
Have fun.
We'll go tour Civil War sites tomorrow.
Have fun.
Yep.
I'm Vincent Racaniello. You can find me at my website, virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.